Hello, and happy Smith-Magenis Syndrome Awareness Day. My name is Dr. Lawrence Ryder. I am a professor in the Department of Neurology at the University of Tennessee Health Science Center here in Memphis, Tennessee. And today I just wanted to talk to you briefly about a project that's ongoing in my lab to collect dental pulp stem cells from kids with a variety of different neurogenetic syndromes, including Smith-Magenis syndrome. So what are dental pulp stem cells? So dental pulp stem cells are cells that live deep inside the tooth pulp of baby teeth and adult teeth as well. And when children lose their teeth, like this kid did here, we can actually collect that tooth remotely within the first 20 minutes, and we'll send you a collection kit so you can collect the tooth and send it back to us, and then we can make dental pulp stem cells from that tooth. We currently have over 200 cell lines in our dental pulp stem cell repository, including 7 lines from Smith-Magenis syndrome. And we would love to have you participate in this project by signing up to send us the baby teeth when they fall out, or even in some cases we've collected teeth that were extracted that were wisdom teeth, and we can use those to grow dental pulp stem cells as well. So once we grow the cells and differentiate them, we can make them into these pretty neurons in a culture dish and use these neurons to study different neurogenetic syndromes. And also specifically, my lab is interested in looking at autism at the molecular level and studying the things that are more common among these syndromes in terms of autism. So this is a study we just published. I won't go into the details, but I wanted you to see that we can actually look at cellular phenotypes within these neurons. In this case, we're looking at this phenotype we found in Prader-Willi syndrome neurons that was a sort of clumping of the mitochondria inside the soma and out into the dendrites of the neuron that's not present in the Prader-Willi kids that don't have autism spectrum disorder. We've also been recently doing a lot of work on circadian rhythm, and we now have data on several syndromes, including a PWS-related syndrome called Chaff-Yang syndrome. And what you can see here is the bright green line is shifted from these other 2 lines. And this is a normal circadian cycle. But in Chaff-Yang syndrome, in this particular cell line, you can see a shift in that circadian rhythm, which could be an indicator of problems with sleep or other circadian regulated behaviors. So what do you guys have to do? Basically, you need to contact me. So my email address is here. And I would prefer you to send me an email, because what I need for participation from you is a genetics report that shows the Smith-Magenis syndrome diagnosis at the genetic level. So if I can get that genetics report from you, I can then send you a kit, and the kit contains everything you need to collect a tooth. It also has some information about autism status, a questionnaire, and then it has a return shipping box and the FedEx return information so that we can get the tooth and we can grow dental pulp stem cells. So I hope everybody is willing to contribute some cells to our study, and it would be great for us to continue to work on Smith-Magenis syndrome at the molecular level and try to understand some more about this syndrome, and specifically when it relates to neurons and how they function. Thanks again.